required, one for the protein, one for the lipid, one for the carbohydrate. And whatever chemical will breaks this protein, we use this word is in the end of this product. So if any chemical which is breaking the protein, we call as protease. Any product which is breaking the lipid, we call as lipase. Any product which is breaking the carbohydrate, we call as amylase. Okay guys, so protein, lipid and carbohydrate are going to break down with the help of protease, lipase and amylase. For carbohydrate we use the word amylo. So the product which is going to be further breaks this carbohydrate we call as amylase, right? So now you can easily understand whatever, whenever, if anybody tells you this is the lipase, this is the protease, this is the amylase, you can easily now understand that this is the protein breaking substance, this is the lipid breaking substance, this is the carb breaking substance, right? So I hope now you can easily understand. So this protein will break, lipid will break and carbohydrate will break. Now please pay attention. Whenever this protein is going to be digested, it is breaking. What is the end product which will extract from this protein? And the end product of this protein is actually your amino acid. From the lipid, it will be the fatty acid and glycerol. And from the carbohydrate, it is your glucose and fructose. Yes, very good guys. So this is the amino acid, then the fatty acid and glycerol, then the glucose and fructose. These are the last substances which are going to be extracted from the protein, lipid and carbohydrate. So whenever carbohydrate will break, this will give last product, that is the glucose, and all these products are now suitable for the cell, and now these products will enter into the cell. So this is how this amino acid will enter into the cell, fatty acid, glycerol will enter into the cell, glucose and fructose will enter into the cell. Okay, now, so protein breaking substance we call as protease, this is we call as lipase, and this substance we call as amylase. Now what is happening? Protein is breaking. Guys, anything breaks inside the body, we use the word lysis. What use the word? We use the word lysis. Okay? So whatever breaks inside the body, we use the word lysis. Like if the RBC is breaking down, hemolysis. This lipid is breaking down, lipolysis. Protein is breaking down, proteolysis. Glucose is breaking down, glycolysis. So this is how all of these conditions we use the word. So this protein breaking functioning we call as proteolysis, lipolysis, and this we call as amylolysis. Amylolysis. Right? So when we'll study the GI part, in GIT I will let you know that this is the protease, lipase or amylase, from which area they are secreting. You just simply add the name before that part. Means if the amylase is secreting from the saliva, we call it salivary amylase, from the pancreas, pancreatic amylase. Similarly, if the lipase is releasing from the tongue, we call it lingual lipase, from the gastric part, gastric lipase, and from the pancreas, the pancreatic lipase. So that is how we are going to use these words. Now this amino acid, fatty acid, glycerol, and glucose and fructose are added. My question is that now, what is the role of this amino acid? What this cell will do with the help of this amino acid? Why this cell is required the amino acid? And why this cell is required the fatty acid, and why this cell is required the glucose. So guys, first of all, let's talk about for the glucose. This cell is functioning like a factory. This factory means, suppose you are having a textile factory, or in this textile you are making some product. In this factory, what you require? First of all, you require the raw material. Then you require the energy part, from where you will get the energy. So similarly for this cell is getting the energy, and this cell is getting the raw material. Guys, this cell is actually having a magic property. And what is the magic property? Magic property that it can divide. Whenever this cell is going to be divided, guys, for division, this cell requires the raw material so that it can produce a new cell with the help of or with the making by some raw material, so that new cell can be developed. And this new cell, or the most of the area of our body, is actually made up of the amino acid. So this amino acid is becoming like the bricks of the life. Like suppose you are making a house, आपको अगर एक घर बनाना है, तो what the most thing you require that is what? Bricks, ईंटें. तो आपको चाहिए सबसे ज़्यादा ईंटें. Now once you get the bricks, using of these bricks, you can make the hall, you can make the kitchen, you can make the bedroom, dining room, all of these areas you can make it. So the most of the part for making the house you require the amino acid. For similarly for this cell, for making any structure inside the cell, this cell is required the amino acid. So amino acid is becoming the bricks of the life. So suppose guys, you are going to the gym. If you are going to the gym, you want to make the body, right? You want to make the big muscles. Big muscle means more cell division, and if more cell division, more raw material is required. And what is the raw material most that is required for cell? That is the amino acid. So that's why after going to the gym, most of the person start consuming more and more amino acid, or in the form of protein. So वो इसलिए बेटा प्रोटीन पाउडर पीते हैं, and coming out from the gym they are eating the 10 or 15 boiled egg. So why? Because the eggs are containing most of the protein. They want to supply over here the good amino acid so that the more and more cell division would be possible. So I hope now you can understand why basically the person needs the protein. Now the next part is the fatty acid. Why the cell is required the fatty acid? This fatty acid is also required to make some area inside the body, because we know inside our body every area is actually present inside the water, because inside the body 60% is water is there. The water which is present inside the cell we call as intracellular fluid. The water which is present outside of the cell we call as extracellular fluid. So means everything will always stay inside this watery environment. But some of the area of our body, this cell don't want to make the water soluble. So that's why all these areas which need to be make the water non-soluble, they can made from the lipid, and that's why we require the fatty acid. So primary structure part, that is the amino acid, and the secondary structure making part, that is our fatty acid. Now what is the role of this glucose? Guys, as we know that this cell is continuously doing like a factory like a job. In this factory you require the energy. Who will give the energy? Energy is produced via the glucose. So this glucose will enter into the cell, it will go into the mitochondria, and this glucose will break into this mitochondria. And if glucose is breaking, what use the word? Glycolysis. So due to this glycolysis will produce the ATP, and this ATP is utilized as an energy form for the cell, and that's why this mitochondria will be named as a powerhouse of the cell. So what is the function of the glucose? Glucose function is to provide the energy for functionality, right? So there this glucose will go inside the cell. Always remember that this glucose from here, it will enter into the cell and will reach into the mitochondria, and that is how you study in the biochemistry that there is a Krebs cycle. And many cycles are there. So that's why only the three metabolism you have to study, that is your protein metabolism, lipid metabolism, and carbohydrate metabolism. So I hope now you can understand. So this is how glucose is required for the primary energy source. Now what happens? Suppose you did the fast, you are not eating the food, आपने व्रत कर लिया. Now what will happen guys? No glucose is entering inside the body. So now from where this cell will get the energy? Because this cell continuously functioning, some of the area of your body, they are continuously functioning, like your heart muscle, it never going into the resting condition. This heart cell is saying continuously provide the energy, otherwise you will stop functioning, and if heart stop functioning, you will be stopped. So how this cell is getting the energy, even you are in a fasting state, guys, this will get the energy from the secondary energy source, and that is our fatty acid. So this fatty acid is also function as is providing the secondary energy source, right? So this is how the different functions of these different food materials. So I hope now you can easily understand. Now talking about this cell, guys, what happens whenever this amino acid from here, whenever this amino acid will enter, where this amino acid will go? this amino acid actually will go near to the endoplasmic reticulum. This endoplasmic reticulum is divided into two, the smooth and the rough. Rough because there is the presence of the ribosome, and this ribosome are the granules-like structure, so that's why this endoplasmic reticulum becoming the rough into the structure, so called as rough endoplasmic reticulum. This ribosome is actually the protein factory of the cell. Once this amino acid will enter into the cell, what will happen? From the information which comes from the DNA, this information tells to this endoplasmic reticulum that you need to make a new protein molecule with using of this amino acid. So that is how this amino acid will enter over here, and then the information will come from here, and this is how the new protein will be formed, which is actually suitable for the cell. So this is how the amino acid is required for the protein synthesis, and this protein factory is actually called as a ribosome, and this ribosome is present at the rough endoplasmic reticulum. So what is the function of rough endoplasmic reticulum? The function is protein synthesis, right guys? Now this information is coming from the nucleus. If information coming from the nucleus, guys, where this rough endoplasmic reticulum should be situated? Nearby to nucleus or nearby to cell membrane? What do you think? Where this endoplasmic reticulum should be situated? Near to nucleus or near to cell membrane? Near to nucleus, right guys? So that is why you can see in this image, this endoplasmic reticulum is always situated near to the nucleus, right? Now after that, this fatty acid, what will happen when this fatty acid will be formed? This fatty acid when formed, it will enter into the cell, and again with the help of smooth endoplasmic reticulum. This smooth endoplasmic reticulum will do the lipid synthesis and will make the new lipid product by the using of this fatty acid. Now suppose this is the production site. Right? Now in the production side, the product is ready. Now where you will send this product? Now you will send this product to the packaging unit so that this product can be delivered out for the cell. So this is how this is the production unit. And now where is the packaging unit? So packaging unit is actually this yellow structure, and this is your Golgi apparatus, right? So what is the function of the Golgi apparatus? Golgi apparatus will give you, or it helps for the packing and then put a label over there and then the delivery. So it is functioning like Amazon, the Amazon app which you use for the online delivery part. So this is the same functioning of this Golgi apparatus. This Golgi apparatus will pack the product, pack the protein, then it will put a label on there, and then it will deliver for the making the new cell. So that's the functionality of the Golgi apparatus. Now when any product is coming from here to here, guys, this amino acid when they makes the chain or when they makes the protein, this protein is making like a chain formation, and this chain could be that much long. Guys, whenever you are sending this chain into the packaging unit, you can't send this chain into like simple in a very lengthy manner. You have to fold this chain, and once you have to fold this chain, you have to put into a bag, and then you can give this bag into the packaging unit, that pack it completely. So the similar functionality is happening from here. Whenever this product is ready over here, this product is going to be full, and that process we call as folding of the protein. This folding of the protein can be done by a special protein called as chaperones, and then this will put into a bag-like structure, and this bag we call as vesicle. What we call this vesicle. So this vesicle is actually present over here. Now this product will present into the vesicle. This vesicle will come to the Golgi apparatus, and now this Golgi apparatus will further do the packing, labeling, and the delivery of this product, and that's why this Golgi apparatus is situated near to the cell membrane. So this is how the different functionality of this different organelle. Mitochondrial functionality, you already know. Now what is the functionality of this lysosome? Why you require the lysosome? Guys, as you can see, this cell is functioning like a factory. So in this factory, the product is making, and if this factory is continuously functioning, in this factory there will be some garbage collection will be there. भैया कचरा भी तो होगा. अगर factory के अंदर माल material बन रहा है तो कचरा भी तो होगा. Where you will dump this garbage from the cell, because you have to always maintain the neat and clean environment inside the cell. So once this cell will get that more and more garbage, this cell need to dump this garbage somewhere, and this is going to be dumped into this lysosome. So this lysosome is actually containing an acid. Once all of this product or all of this garbage dump into this lysosome, all this garbage are going to be burned with the help of the acid, and that's why this lysosome is becoming the garbage collector of the cell, right? Or also we can say the suicidal bag. Why? Because during the starvation condition, what is the starvation condition? When this person is not eating any food for this long duration of the time, only during that time, from the non-essential part of the body is going to be killed, and this protein, this lipid or glucose will supply to the essential area of the body, and this killing will be done by the lysosome. So that is how this lysosome is also becoming the suicidal bag, and also this will be seen during the starvation, and this process we call as when cell lysosome eating or cell eating itself only, we call as autophagy, right? So that we call as autophagy. So this is how the different functionality of this different organelles. Now the next part, that's something called as cytoskeleton, as we have mentioned over here. That organelle name is cytoskeleton. Guys, cyto means cell, cyto means cell, and skeleton means bones. Guys, what is the meaning of skeleton? Cytoskeleton, cell, bones. What do you think? Cell does contain any bones? Does cell contain any bones? What do you think? Does cell contain any bones? Yes, guys. Cell can also contain the bones. What is the function of the bones inside the body? Just let me know. Why you require the bones? This bones actually providing the framework of your body, so that all the organs they should place at their proper area. Like your heart should be present into the thoracic area, your kidney should be present at the retroperitoneal area, right? So this is how all of these structures of your body are actually placed at a proper area, and this could be done with the help of the skeleton system. So similarly, inside the body or inside the cell, that's this organelle which is situated inside the cell, they must present always into a proper certain area. Like nucleus should always present into middle of the cell, the Golgi apparatus should always present to the nearby to the cell membrane, and who is holding them? This could be held via with using of the bones of the cell. But as we know, the cell is very small in size, so bones could not be made up from the calcium, so could not be very much of part. But these bones are actually looks like the threads, and these threads are actually three type of threads are there based on their size. The number one thread which is very bigger in size, number two which is intermediate in size, and number three which is very smallest in size. And these threads we call as filament. What we call as filament, right? So the filament which is very bigger in size, So if any pipe which is very bigger in size, this pipe should be hollow in between of this. बिल्कुल खोखला होगा guys, पूरा solid नहीं हो सकता. So the similar part, the biggest fiber is going to be hollow in between of this, and making this hollow, this thread is looking like a tube, and this tube we call as microtubules. So the first cytoskeleton, that is our microtubule. Then number two is intermediate filament, and number three is microfilament. So three types of cytoskeleton is there, and they actually provide the framework of the cell, and they hold all the organelle at their proper area, so that no organelle can go from one area to another area, right? So this is the functionality of the cytoskeleton. So please write it down about the cytoskeleton. 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 This is the bone of cell divided in three types. Vishnu, thank you for your compliment. But I am just the teacher. I am just trying to explain this video in a very easiest manner. If you are liking, then it's okay, and this also will give me the more energy to teach you in a more simplest way. This cytoskeleton is three types. Number one is your microtubule. Microtubules, which is biggest in size. Then number three. Number two is intermediate filament. Intermediate filament. They are most abundant. Most abundant का मतलब होता है very much, बहुत ज़्यादा. So they are most abundant. Number three is microfilament. Number three is microfilament. So microtubules, intermediate filament, and microfilament, and they are the smallest in size. They are the smallest in size. Now, as we know that inside the body, everything is made up of either the protein or either the lipid. The most of the part of the body is made up of the protein, right? Because we know that body is consuming only three parts, that is the protein, lipid, and carb. Carb is utilizing for the energy source. The protein, which is the most abundant source for making the body structures, and then also body can use the lipid. So right, so that's why in the body, the most of the area is made up of the protein, and if that protein is going to be defect in the body, that's we are calling as a defective part. And that's why in the medicine or in pathology, many times we study that this protein deficiency, this protein is defect. That's the reason behind it, right? So this microtubules is made up of the protein, and the name of this protein is tubulin. Tubulin. This is the tubulin. Then intermediate filament, this is made up of desmin protein or the vimentin protein. Vimentin protein. And this microfilament is made up of actin, myosin protein, actin and myosin protein. This tubulin is actually helps. This tubulin helps during cell division. This helps during cell division. Cell division. So this is the three cytoskeletons are there. I hope you noted. Now let's write it down. The functions of different organelles. So number one organelle, that is our nucleus. The function is it basically provides the DNA storage. This nucleus, it is absent in RBC. This is absent in RBC. Then the smooth endoplasmic reticulum, SER. The function of smooth endoplasmic reticulum is lipid synthesis. Lipid synthesis. Then there is detoxification. Also, steroid hormone synthesis. Steroid hormone synthesis. And also it provides the calcium storage. Calcium storage in skeletal muscle. It also provides the calcium storage in skeletal muscle. Guys, this MS means muscle. Right? So all the time I will write something like that. So please be understandable that this means the muscle. So calcium storage in muscle, skeletal muscle. Later on I will let you know that's why we require this calcium, why we are calling that this is the providing the storage part. Okay. Then the rough endoplasmic reticulum, the function is protein synthesis. Protein synthesis. And this protein synthesis, this we known as translation process. Translation process. Translation process. This T and N means TION. So translation process. The folding of the protein, because once the protein is ready, we require to fold this. So folding of the protein done via the special protein named as chaperone's protein. Chaperone's protein. And this happens due to which organelle? Ribosome. This is due to ribosome. This is due to ribosome. Guys, suppose this folding of the protein, suppose this is the chain, if this chain need to be folded, suppose this folding could not be possible. What will happen? Could not be possible this folding. Now, this protein we call as misfolded protein. This misfolded protein, cell don't want to send it out from the endoplasmic reticulum, because if suppose in a factory, if any product gets defective, what will happen? You don't want to send it into the market for the selling part. You will again immediately identify this defective product and immediately send for the recycling. The similar things happens for the cell. This misfolded protein, if start happening inside the endoplasmic reticulum, this immediately identify via the endoplasmic reticulum, and this identification could be done via a marker protein. This marker protein named as ubiquitin. So this ubiquitin is attached to this protein, and once this ubiquitin will be attached, this is giving a signal that this protein is misfolded protein. This destroy this misfolded protein, and this destroy or this degradation could be done via ERAD, endoplasmic reticulum assisted degradation. If this degradation could not be possible, this misfolded protein start coming out from the cell, but none of the cell will utilize this misfolded protein. Ultimately, this misfolded protein is going to start deposit inside of our body, and if suppose they are going to get deposit into the CNS region, we call this Prine's disease, right? So this is your Prine's disease. So please write it down. If the folding of the protein, if misfolded protein forms, it is degraded via ERAD. It is degraded via ERAD. ERAD stands for endoplasmic reticulum assisted degradation. Endoplasmic reticulum assisted degradation. endoplasmic reticulum assisted degradation, and the marker protein is ubiquitin. Marker protein is ubiquitin. Right? Marker protein is ubiquitin. If misfolded protein doesn't degrade into cell, they starts depositing